where state-of-the-art DNP experiments are performed, even lower than the standard DNP experiments, as you will see in the talk. So just a brief introduction to the paramagnetic effects before we go into the details. Whenever the basic idea of the paramagnetic effects are whenever there is a paramagnetic center present in a solid state or in an NMR sample, then the nuclear spins that are located close to the paramagnetic center, their NMR properties gets influenced by the presence of the hyperfine coupling between the electron spin and the nuclear spin. And there are two major implications of that on NMR spectra. One is known as paramagnetic relaxation enhancement, known as PRE, which basically means that both relaxation rates, T1 and T2 of nuclear spins become faster, and that means the line widths of the NMR spectra becomes broad, as shown here by this cartoon, that the nuclear spins that are located far away, they can still have sharp lines, but as you get close to the electron spins, the lines become broad. Now, the second type of effect is on the chemical shifts of the nuclear spins, which can originate from either the isotropic part of the hyperfine coupling, Fermi contact interaction, or the anisotropic part, which is standard dipolar coupling through space. The two types of effects are known as chemical contact shift or pseudo contact shift. And in both cases, the peak position will be shifted from its isotropic chemical shift. Now, these effects can also be anisotropic, and I'll not go into the details, but in that case, the chemical shift anisotropy pattern will also change. In literature, both the PRE and the pseudo contact shift effects have been utilized extensively for gathering structural information in both solid state and solution state NMR. However, under DNP conditions, it can be that sometimes these effects may cause some complications in NMR spectra. So just a brief introduction to DNP. This is a technique, probably most of you are already familiar with it, that is being rapidly used nowadays to enhance the nuclear signal, NMR signal by orders of magnitude. And the basic idea is in NMR sample, we introduce a source of paramagnetic center. Usually these are stable nitroxide radicals, and we mix it with a DNP solution, basically a glass forming agent, so that that acts as a cryoprotectant as well as provide this proton spin bath. And then we irradiate microwaves on the optimum frequency for DNP near EPR frequency of the radical, and that leads to polarization transfer to nearby protons. And because proton spin coupling network is strong, the spin diffusion among proton spins takes the polarization to large distances from the paramagnetic center, and finally we can use standard cross polarization to enhance the signal of the target spins. And this leads to a huge enhancement in NMR signal and a significant time saving that allows us to do new science nowadays. However, one should here in this case, the focus is that we introduce this source of paramagnetic center in the NMR center in the NMR sample, and that will have some side effects, which is known as those paramagnetic effects I briefly discussed in the last slide. So here are some examples where these paramagnetic effects under DNP conditions have been reported. And first example comes from Bob Griffin's lab, where Professor Corzelius has shown that if you increase the concentration of these biradicals or the trital radical or the monoradical or even the gadolinium DOTA, what we see is the relaxation rate of, in this case, they observed carbon-13 transverse relaxation time increases, and that results into line broadening, and hence the spectral analysis can be complicated. And more recently, group of Professor N. McDermott showed that if you attempt to perform DNP with some tethered biradicals to the target system, which can lead to more precise information, not only the signal enhancement, but you can get direct distance information as well. But in that case also, they observed that some of the signals, those are highlighted in red here, they quench because they get too close to the paramagnetic center. That means their relaxation times becomes too fast, and the lines broadens out. Now the question is, can we deal with these effects? Is there some possibility of paramagnetic effects reversal or suppression? And there have been attempts in both from electron nuclear decoupling. However, I will not go into that today. I'll show you some of the interesting effects that we saw by microwave irradiation on these paramagnetic effects. So the system that we used was a lithium electrolyte solution, and we mixed it with tempo methacrylate as a radical for DNP to detect DNP enhanced lithium NMR signal. And the experimental conditions are shown here. We perform experiments at liquid helium temperature. Our field is at around 7 Tesla, which correspond to these frequencies for proton and electron spins. So here is a comparison of the lithium NMR signal recorded without any radical in. So that's a blue line, which is relatively narrow. And as we introduce the radical, the line becomes broad and its chemical shift changes as expected. Now, to our surprise, when we try to do this DNP, which means we radiated microwave for build-up time TDNP, which is around 60 seconds, and then we recorded the DNP enhanced NMR signal. What we observe is that there was signal enhancement, but I'm showing here the normalized signal to highlight the fact that the lines are narrow, the line become narrow now, and the chemical shift reversed back towards the original position. So this raised the question that is it some kind of reversal of paramagnetic effects caused by the microwave irradiation? However, one needs to address these questions that it could be just microwave heating because it's ultra low temperature. So irradiation of microwaves can be absorbed, and that could lead to some line narrowing. However, we are not sure if that would also lead to reversal of the chemical shift. It could be an actually electron and nuclear decoupling because the microwaves are on throughout the experiment. So during acquisition, maybe the microwaves are leading to some decoupling of the electron nuclear hyperfine coupling. It could be selective DNP, and by that I mean some of the lithiums which are located slightly far away so that they are not influenced by the paramagnetic effects are getting enhanced more than the other ones that are more affected by paramagnetic effects, and hence we are observing the signal which look like it's reversal, but it's only this selective DNP. So to analyze these and to answer these questions, we need a system where we can have full control on our microwave capabilities. That means we would like to have a system where we could stop the microwaves whenever we want. We can change the microwave frequency or power level. And for that, we used our home built system, which provides relatively low power. It has been upgraded now. And so now we have almost 400 milliwatt power. But at the time of these experiments, we could have 120 milliwatt microwave power in a broad frequency range of 10 gigahertz, around 194 gigahertz central frequency. So the main features of this system is we have this cryostat which allowed us to do achieve liquid helium temperature. We simply fill it with liquid helium so to achieve 4 Kelvin. That has been also upgraded now to helium-free cryostat. Then we for the microwaves, we have a microwave source, and that the microwaves go through a quasi optical system and then irradiate on the probe that is equipped with corrugated waveguide to transmit them to transfer the microwaves to the sample. So this system has an advantage that in this we can rapidly switch the frequency of the microwaves. The tunable range of microwaves is quite wide, as I said, 10 gigahertz. And most importantly, we can pulse microwaves. We can stop microwaves within nanoseconds, and we can shape it if needed. We can modulate the frequency during the experiment. And we have EPR detection capabilities along with the NMR detection capabilities. For the RF part, we simply used a very simple single resonance probe that was also home built, and it's based on inductively coupling, which means there is no physical connection between the picking coil and the sample coil. So with that, now I will go to the results, and first we try to explore whether it's a temperature effect. So we varied the temperature during the experiments. So the parameters are shown here. The irradiation time is 60 seconds, which is needed for polarization to build up, and that is dominated by lithium NMR relaxation time. The microwave power was used at its maximum, 120 milliwatt. The frequency was 193.7 gigahertz, which is the frequency at which the DNP was optimum. We also tried a different frequency, which is at the center of the EPR pattern of the tempo at our field. So the black data points here are the microwave off as a function of, and the line widths are line widths as a function of temperature under microwave off conditions. And we see that as we go to 10 Kelvin, 15 or 20, the line widths decrease. So of course that higher temperature leads to narrower lines. But when we irradiate it with microwaves, the lines become even narrower. Now one could say that it could be still the temperature effect because the irradiation of microwaves are changing the temperature. First of all, this would require a temperature change of 6 Kelvin because here are the comparison of 10 Kelvin and 4 Kelvin with microwave. So microwave should increase the temperature 6 Kelvin, which seems highly unlikely. But more importantly, what we see is that the effect of microwaves is frequency dependent. And we are changing the frequency only by 200 megahertz, and even by that small change, the line width is significantly changed. So it doesn't seem like it's a heating effect because heating should not depend that much on the microwave frequency, especially it's only 200 megahertz away. So then we went to the next parameter, and now we try to use our pulsing capabilities. So first of all, we removed the microwave irradiation. We stopped the microwave irradiation during acquisition. And if it was a decoupling electron, the standard decoupling that we use in solid state NMR or in solution state NMR, then the lines should be broadened now because during the acquisition there is no irradiation. However, we didn't observe that. The lines were still narrow. In fact, we tried to vary this build-up time, and what we observe is that the lines get narrower quickly within 1 second. Within 1 second of the irradiation time, whereas the polarization buildup is a much slower process. And I'm not showing the polarization buildup curve here, but basically it takes around 60 seconds, as I mentioned earlier, to build up the polarization. So the two processes, the building up the nuclear polarization and the line narrowing of the NMR spectra are occurring at completely different rates, which tells us that it's not the selective DNP most likely, because that should have been at the same rate as the polarization buildup. So then we try, in order to understand it in more detail, we try to vary the other microwave parameters. And the next microwave parameter that we varied was microwave power. So the microwave power is proportional to the square of the microwave power is proportional to the nutation frequency of the EPR spins caused by the microwave irradiation. And the red data point here is the DNP enhancement as a function of microwave power. What we see is around 80 milliwatt, the signal enhancement is maximum, around factor of 20. And the line widths are linear, monotonically decreasing as we increase the microwave power. Now, what we see here is that at higher power, beyond 80 milliwatt, the DNP signal then starts to decrease. And this is a phenomenon known as oversaturation that has been reported by our group earlier. And this is an important hint towards electrons spectral diffusion that results into such behavior, which means that the EPR pattern is the EPR pattern is getting quenched by microwave radiation quite effectively. So just to see this effect directly, we could use our EPR detection capabilities now to see exactly what's happening on the EPR side, on the electron spins, while we are performing DNP experiments or irradiating microwaves. And for that, we used a standard EPR experiment known as ELDOR, electron-electron double resonance experiment. And in this experiment, the basic idea is we irradiate the sample at one frequency while detect the echo at another frequency. And we vary the pump frequency, the frequency at which we irradiate. We vary the pump frequency while detecting at a single probe frequency. And the idea is if the two spins are coupled directly or through a third spin or fourth spin in between, then the signal will, the saturation will be transferred from the irradiation frequency to the probe frequency, and that would lead to a pattern that looks like this. So this is basically a saturation profile of EPR pattern. And a broad saturation profile, a broad hole burning in the EPR line width tells us that the EPR line is homogeneously broadened and a prominent spectral diffusion is present. If it's a narrow hole burn, then it's a inhomogeneous line, and that means that spectral diffusion is not that effective. So what we observe is that within 100 milliseconds of the pump pulse, which means very short microwave irradiation compared to the DNP buildup time, which is in 60 seconds, the EPR pattern is significantly quenched. There is a broad hole burned in the EPR line width. And this tells us that this is also a fast process, the saturation of the EPR pattern, which is in agreement with what we observed in the line narrowing effect. That line narrowing effect is significantly faster than the DNP buildup. So it seems like from these parameter variations and observing these effects with function of microwave time, irradiation time, power, and EPR saturation, that this is not microwave heating. This is not active electron nuclear decoupling, because we don't need irradiation during the acquisition. It's not selective DNP, because the rate of the line narrowing is much faster than the signal buildup, DNP buildup. So it could be, it seems from here that the saturation of the electron spin polarization is somehow influencing the correlation time. And by correlation time, I mean the electron spin relaxation time. So I'll briefly introduce this theory, which most of you are probably already familiar. But in standard solid state and solid state NMR, we use these expressions to determine the PRE effects on both T1 and T2 relaxation times of the nuclear spins. Now, these theories are valid at high temperature, where the correlation times are quite fast. At low temperature, I should point out that these theories needs modification and their full understanding is still not there. But it has been reported in literature that the correlation times here are dependent on the electron spin relaxation times. Which means that if we go to low temperature, the rotational correlation time and the chemical exchange correlation times, they become irrelevant because they are too long. And the dominating factors in the correlation times become the electron spin relaxation times. And in fact, there are studies where they show that if you go to really low temperature, where electron spin polarization tend to be close to 1, then it's the T2 of the electron spins that become the most influential factor. So then the question is, are we changing the relaxation times by irradiating the microwaves on the electron spins? So to test that, we ran the similar experiment while measuring the electron phase memory relaxation times. And phase memory relaxation time, I should mention here, is similar to T2, but it's a overall effect on the decoherence of the electron spins. So it is sort of T2E of the electron spins. So here are the results. The blue data points are the line widths as a function of microwave power while decoupling, while doing the DNP experiment. And the black data points are recorded using this pulse sequence on electrons where we irradiate with the same parameter as we used in DNP on one frequency, on the frequency where we irradiate for DNP, and then detect at the center. And what we observe is that the phase memory relaxation time, the T2E of electrons, also follow the same trend that also decreases in the same manner as the line width. Now the next parameter that we varied is the time of this pump pulse as we vary the TDNP, the build-up time, and we observe the similar effect. That means the electron phase memory relaxation time is also reduced quite rapidly within few milliseconds, hundreds of milliseconds of the pump irradiation of microwaves, which is similar in what we observe on the line width pattern. And the same effect we have observed on the chemical shifts as well, which I'm not showing here, but you can see in this paper where we show that both chemical shifts, pseudo-contact chemical shift and contact chemical shift, also follow the same trend. So from these experiments, we see a strong correlation between the change in the TM as a function of microwave parameters and the change in the paramagnetic effects as a function of the microwave parameters. So with these results, however, it's not completely confirmed that it is the reason, but it seems like that the saturation of the electron spins can provide, and detecting the electron spins can provide more insights into the underlying mechanism of these paramagnetic effects under these conditions that is extremely, that is ultra low temperature.
So Sheetal, you have a couple of more minutes.
Yes. So, so finally, what we can do with this type of understanding and what we can achieve for DNP experiments. Basically, we have seen that we don't need irradiation during the decoupling time, during the NMR signal acquisition. However, we know that if we put a small block, because the electron spin saturation does not require a long irradiation time. So if we can switch the frequency right before detecting the NMR, we could achieve so-called narrow lines. At the same time, we can build up our signal before this long irradiation at optimum frequency. So basically, in this case, I'm showing you the results where we vary the microwave frequencies during both the blocks with this, while keeping the both of them same. What we see is that this is the standard DNP profile, but here, the line widths are minimum when the DNP signal is almost zero. The DNP enhancement is also almost zero. So in order to achieve both of them, we should be irradiating here while DNP build up and irradiating here while decoupling or this saturation, so that we can achieve maximum of both. And that's exactly what we did. If we now fix the microwave frequency here to the maximum of the DNP, we observe that the signal is maximum, while if we vary only during this block, then the line width becomes narrow as we hit the center of the APR. And another implication of this is that if we now fix the irradiation during this small block to be at the center of the APR frequency, and then record the standard DNP profile, we see no effect on the DNP signal, but the lines are now narrow. So the basic idea here is that by this frequency switching capabilities, we can get narrow lines without compromising on the DNP enhancement. So with that, I would like to summarize that these pulsing capabilities allowed us to get some insight into the paramagnetic effects, and the implication of that is recording DNP enhanced NMR signal with reversal of the paramagnetic effects that has that leads to line narrowing in NMR signal. With that, I would like to thank my supervisor, Professor Songi Han, and the collaborators who contributed to this work, the funding agencies, and the Han lab here. Thank you. I'll be happy to take questions now.
Thank you, Sheetal, for finishing in time. So the talk is now open for questions. You can raise your hands if you have any questions. I mean, virtual hands. Raise your virtual hands. Yes.
Vipin has a question.
Vipin? Yes, Vipin.
Yeah, not able to see how to raise my virtual hand, but if I'm audible, may I go through with my question?
Yes, please.
Yeah, I can hear you.
Yeah. There are two things. If I remember right, you said that the build-up time is of the order of a few seconds.
Yes.
You did not quite mention, unless I failed to catch it, what is the time duration of the frequency shifted block?
It's 100 milliseconds, 100 milliseconds.
So it is either 10% or considerably less than the irradiation time for the buildup.
Considerably less. The build-up time is 60 seconds.
Okay, great. So 60 seconds versus 100 milliseconds. Okay.
Yes.
Now, how does this kind of findings, how do they relate to some work which was published about 3 or 4 years ago, which also showed a line narrowing of the NMR signal under solid state DNP conditions?
Yeah, so that was by Professor Jenderbahn's group, I think that's what you're referring to.
Mm-hmm. This was about 3 or 4 years ago.
Yeah, so in that case, so if you notice here, we do not put microwave power during the acquisition. So this is not the active decoupling. In that work, they actually did electron nuclear decoupling by irradiating during the signal NMR detection, and they varied the microwave frequency. So it's different from that, mechanism wise different.
Agreed, but the question is, from the point of view of the end result of the DNP, is there any merit in combining these two approaches? Although you claim that irradiation of microwaves during the acquisition of the NMR FID doesn't seem to make a difference.
Yes.
Seems to be some bit at odds with the earlier findings that I'm talking about. And therefore the question is there a prospect of combining these approaches? You made a very good case for the correlation time of the electronic motion being a function of the T2E or the T1E as the case may be, and that being the main source of the influence you're seeing. But then how does it mesh with the other kind of finding?
Yeah, so can we combine the two? Because the two effects are in completely different regimes. The earlier effect, the decoupling, was shown at 100 Kelvin. What we showed here is at 4 Kelvin. And this means, in a way, this is more restricted because this requires this saturation of the electron spins. However, one can combine the two because in the work that was shown by Professor Barnes, they used frequency agile gyrotron, which means they modulated the frequency of microwave to saturate the EPR pattern. And that actually can be combined with it. We didn't do those kind of experiments yet, but certainly that will be interesting to see.
Okay, thank you. Thank you for a very nice talk. Thoroughly enjoyed it.
So Vipin, you can...
Hi, can you hear me? Hi, Sheetal, very nice talk. I was just wondering, how is this electron TM different from the classical T1 row kind of experiment that one measures? And that's the first part. And the second part is, if you play around with the properties of this TM block, then how does the T1 and T2 change?
Really good questions. So first thing, the TM is ideally, when normally we record TM, we don't need this pump pulse. It's basically this two pulses, and you record the echo and you vary the delay. So it's basically T2. But we call it TM because in electron spin dynamics, there are a lot of factors that influence it, and it's a terminology used in EPR, but it's very close to T2, what we see in NMR. Now, the second question that how will, if I use this, if I vary this, how the T1E changes, we did not see any change in T1E. And that's because T1E, this is basically saturation in that case. So it will be basically saturation recovery experiment, and changing the saturation efficiency will not change the T1E. It will just make it the data more reliable or less reliable depending on how effectively you saturate. But the T1E was not, at least to a significant extent, was not influenced by this.
But that sort of contradicts the argument, right? Because you, on one hand, say that the correlation time changes. And if your correlation time changes, then your T1E has to change, right?
Yeah, so so exactly, you are right. The point, so that's why it was surprising to us. And that's why we make a case that in this case, in this regime, the correlation time is not dominated by T1E, but by T2E. T2E is changing, T1E is not changing. So that's what I showed in this slide here, that the correlation time is not only T1E, but also T2E. And if you go to a regime where the polarization becomes almost 1, then there are studies that has shown that T2E can be more relevant to the.
Okay, so how different are the T1E and T2E in terms of magnitude? Because that would define what should dominate.
Yes, you're right. And T1E, and I should have mentioned it, sorry about that. T1E is in hundreds of milliseconds in this regime, whereas T2 is in microseconds. So they are completely different.
Okay, thank you. Thanks.
Hundreds of milliseconds of T1E, did you say?
Yes, because we are at 4 Kelvin.
Sorry? Yeah, sure. Yeah. But then hundreds of microseconds for the T2E. So there's kind of 3 orders of magnitude there.
Yeah.
Okay.
All right. So thank you, Sheetal, for a very exciting talk. So may I request you to now stop sharing your screen so that we can move to the second talk?
Sure.
So second talk is by Professor Rina Rosenzweig. So Professor Rina Rosenzweig, brief introduction about her. So she completed her BA in molecular biochemistry at Technion, Israel Institute of Technology in 2003, and then she completed direct PhD program there in 2010. She then moved to University of Toronto, where she studied molecular mechanisms involved in guiding protein refolding and disaggregation. And then she joined Weizmann Institute of Science in 2006, where she's using NMR to understand molecular mechanisms that can rescue cells from the accumulation of misfolded and aggregated proteins associated with neurodegenerative diseases. She has various awards, and the latest one I will read out, the Israeli Foundation Faculty Fellowship Award. So with this brief introduction, I welcome Professor Rosenzweig to discuss about molecular chaperones in protein disaggregation. The floor is yours.
Thank you. Thank you so much for the kind introduction, and thank you so much for inviting me. I would like to switch gears and talk about one project that we recently finished in the lab. And we do NMR application, and we study molecular chaperones, especially molecular chaperones that are involved in protein disaggregation. Now, why do we care about chaperones? Well, first of all, because these are absolutely essential proteins that help proteins to gain their proper functional 3-dimensional native state, but also because chaperones are very important to making sure that once proteins are folded, they will maintain folded, which is very important in a crowded environment of the cell, and especially upon stress. And this is important because misfolded proteins are, of course, cannot do their function, but also because the misfolded proteins have the tendency of sticking together and forming protein aggregates and amyloid fibers, which are a hallmark of many misfolding and neurodegenerative disorders. Now, for many years, it was thought that once those aggregates and amyloids are formed in human cells, this is it, this is a dead end, and they cannot be restored back to their functional native states. And it's about 5 years ago that it was discovered that this is not the case. And we have certain molecular chaperones inside our cells capable of breaking apart these aggregates and amyloids and refolding the proteins back into their fully functional native states. And what I would like to talk to you about today is the chaperones that are involved in the segregation of amyloid fibers. And these were described at the end of 2015 by a group of Bernd Buchau, our collaborators. And they found that HSP70 chaperones, together with a very distinct co-chaperones, can solubilize amyloid fibers. Now, with years, it was shown that they are functional over a wide range of various disease-associated amyloids, like, for example, alpha-synuclein fibers, Parkinson, poly-Q fibers, and tau tangles found in Alzheimer's disease. And you can see here negative stain EM images of the fibers. And once just those three chaperones are added together with ATP for energy, this is all done in vitro, within hours, those fibers are completely dissolved and the proteins are back to being monomeric again. So this is a really efficient disaggregation machinery. And we were really interested in understanding how this machinery works, how these three chaperones, HSP70 together with the two co-chaperones, can break apart with very stable structures of amyloid fibers. And what we asked as our first question is, we know that we have HSP70 and we know that we have HSP40, also known as J-domain proteins. And in order for protein disaggregation to occur, these have to be of class B only. And we wanted to understand what is so special about those co-chaperones of class B. Now, J-domain proteins, there are about 50 different J-domain proteins in humans. These are also known as the HSP40 family. They are divided into different classes based on their different domain organization, but they're all known to do two functions. They are co-chaperones in HSP70, and what they do is they bind and deliver substrates to HSP70 chaperones, or bring HSP70s to certain locations in the cell. And therefore, these J-domain proteins are responsible for really many functions that the HSP70s have in the cell. But also, they are very important regulators of the HSP70 function, because when the J-domain proteins will bind to HSP70, well, they will activate its HSP hydrolysis rate, and HSP70 chaperone will convert from an ATP state, where it has low affinity for substrate, to an ADP state where it has high affinity for substrate, and then it will bind to the substrate. It can remodel it, partially unfold it, and help it gain back its functional state. Now, in order to substrate to be released, there is an additional player, nucleotide exchange factor, that releases the ADP and exchanges it for ATP, and this has to be HSP110 for protein disaggregation. So, looking at these J-domain proteins, and knowing that only class B are functioning protein disaggregation, what we wanted initially to understand is what is so special about those class B J-domain proteins, why only they can work with HSP70 in protein disaggregation. And we thought if we can understand that, this will give us a first clue about how this protein disaggregation actually works. And this is a particularly intriguing question if we compare between two known classes, class A and B J-domain proteins. You can see their domain organization here. Structurally, they're very, very similar proteins. But class A with HSP70 do not disaggregate at all, while class B are fully functional. Now, what you see here is both class A and B are homodimers. They have this dimerization domain followed by two substrate binding domains. So this C-terminal domain 2 and C-terminal domain 1 in cyan. Class A also have this zinc binding-like region, which is absent from class B. So this is the difference between the two classes. These domains are responsible for interacting with substrates. And in addition, we also have this N-terminal, very, very conserved J domain. And this is the domain that binds and activates HSP70. And this domain, it's connected by a long, flexible glycine-phenylalanine-rich linker to the rest of the protein. So there is no crystal structure of the whole protein, only of the substrate binding domains and the N-terminal J domain. So looking at this and knowing that the functions of the J-domain proteins is to bind substrates for these substrate binding domains and to activate HSP70 for this J-domain, we initially thought that maybe the difference in their disaggregation activities stems from the fact that class A cannot bind to the amyloid fibers. Maybe because of the different architecture due to the presence of the zinc binding domains, but class B can. So we started by testing that. And we used class A member DNA J2 and class B member DNA JB1. And we tested their binding affinity to alpha-synuclein fibers characteristic of Parkinson's disease. And we found that, in fact, both A and B bind to the fibers with pretty much similar affinity. And yet class A shows no disaggregation activity. So since the binding to the substrate seems to be the same, we were left only with the fact that the two must somehow interact differently and activate differently the HSP70 chaperone. So we then moved to testing the binding of the two chaperones to HSP70 by NMR, and we decided to use a small construct having this domain of the internal domain, that the J domain that is known to bind and activate HSP70, and we also decided to include this glycine phenylalanine rich linker. And the reason we wanted to use it because there were some reports in the literature saying that this GF region might be important for interaction with HSP70. And class B actually have a much longer GF region. So we thought maybe this GF region in class B binds and helps to activate HSP70 more for class B compared to class A. So we started by looking at, by NMR, at this construct, first class A, containing the J domain and the GF. We add HSP70, we see nice peak broadening, the binding is intermediate exchange. We can plot the intensity ratios and map the binding onto the structure of DNA J2 J domain GF. And we see that HSP70 binds to helix 2. It has this conserved HPD motif here, which is also involved in binding. Biochemically, it was shown to be essential for the activation. And then we see that also HSP70 binds to helix 3. Now, the GF region is not affected at all. The disordered region does not play a part in the interaction. So then we move to class B. This is the spectrum of DNA JB1, J domain GF. Again, it had the longer GF rich region, and we thought that this time maybe the GF will bind, and we will see some difference. But when we added HSP70, we were really surprised to see that there is absolutely no interaction. And this was particularly surprising because if we now repeat the binding experiment using the J domain of DNA JB1 without the GF region, we get a really beautiful binding. So there is something about this GF disordered region that somehow abolishes the ability of the class B DNA JB1 J domain to interact with HSP70. So then we got curious and we wanted to study this construct by NMR. And we started by determining the secondary structures. So we had all the assignments, we determined the secondary structure, and we saw that the construct has 4 helixes. Now, 4 helixes were known. They're coming from the J domain, and there is a structure, the NMR structure for the J domain. But we were surprised to see that the GF region, which up until that point, everybody thought is completely disordered, is actually only partially disordered. And towards the end of it, it has a very stable helix, which we call helix 5. And we can also measure order parameters for this protein, for this construct. And what we see is, again, we see the stable ordered region of the J domain of the 4 helixes. Then we see the disordered part of the GF, and then the helix 5 seems to be part of the J domain. So it somehow must be interacting with the J domain. So at that point, we were curious enough to try to solve the structure of this DNA JB1 J domain GF construct by NMR. And our initial idea was that we're going to use CS Rosetta. So we have all the chemical shifts, we added some PREs, and we tried to get the structure. But what happened is that we got a really nicely converged structure of the J domain containing the 4 helixes, but the GF region did not converge at all. So in order to get the structure of DNA JB1 J domain GF, we ended up collecting quite a lot of PREs, NOEs, and RDCs from 2 fields in order to get an NMR structure. But once we have the structure, everything became really clear. So if we now compare the structure of DNA JB1 J domain GF to that of the J domain, we now can understand why HSP70 cannot bind. So if we take our NMR data and map where HSP70 interacts on the J domain of DNA JB1, these would be the blue regions, which contain helix 2, the HPD motif, and helix 3. So this is the exact binding we saw for class A J domain proteins. So this interaction seemed to be conserved between the two classes. But now, if we look at the construct that contains the GF region, we see that the helix 5 of the GF region actually docks onto the J domain on helix 2 and helix 3, blocking the HSP70 binding site. So in the presence of the GF-rich region, which is not disordered as we previously thought, it forms this helix 5 that docks onto the J domain and prevents HSP70 from binding. And this seems to be very different between the two classes, the A and B classes of the J domain proteins, where in class A, we have the GF region not participating in the interaction and not affecting the interaction at all, while in class B, the GF region seems to be blocking the interaction and playing some regulatory role. Now we know it has to be some kind of regulation, because for many, many biochemical experiments, we know that if we take DNA JB1, full-length protein, and we add it to HSP70, HSP70 will be activated. So we know that in the full-length proteins, this inhibition must somehow be lifted. So to understand how this regulation works, we wanted to look now by NMR on the full-length DNA JB1 protein. Now, DNA JB1 is an 80 kilodalton protein, and it's also in homodimer. So we needed to use the methyl labeling in order to see the NMR spectrum of this protein. And you can see here the isoleucine-leucine valine spectrum. And we know from the position of the chemical shifts that in the full-length protein, they have this helix 5 forming, and we have the J domain inhibited. But yet, when we added the HSP70 chaperone, we can see again peaks disappearing in intermediate exchange, and meaning that there is binding, meaning that the inhibition is lifted. And once we obtain full assignments for DNA JB1, we can map the interaction, and we can see that first of all, we see binding to the J domain, but also we saw a new interaction of binding of HSP70 to the C-terminal domain 1 of DNA JB1, which is the substrate binding domain. C-terminal domain 1, the cyan residues here. So not only the GF inhibition was lifted, we also saw by NMR that we have two binding sites, one expected at the J domain and another one new in the CTD1 region. So the next step for us was to take HSP70 and look by NMR from HSP70 sites to map where our two binding sites occur. And doing that, we found that the J domain of DNA JB1 interacts at the interface between the nucleotide binding domain and substrate binding domain of HSP70. Now, this interaction is very, very similar. There is a crystal structure of E. coli HSP70 DNA K in complex with E. coli J domain of DNA J, and the interaction occurs here. So this interaction seems to be conserved from bacteria to human, but the new binding site in CTD1 actually interacts with a very C-terminal disordered tail of HSP70. So we see that there's two binding sites, and we wanted to see if this second binding site, the interaction between the C-terminal HSP70 tail and CTD1, is also present in class A J-domain proteins. So class A J-domain proteins, they lack the GF inhibition. Now the question is, do they have the second binding site? And we were very lucky because the C-terminal tail ends with the EEVD, which has a valine, which we can see in our methyl labeled NMR spectrum. So this is valine here, valine 645. It gives a really, really intense peak because it's at the very C-terminus of the protein. And if we add DNA JB1, you can see that this peak is completely gone because it's interacting with DNA JB1. But now if we do the same experiment and look at labeled HSP70 in complex with DNA JA2, we can see that the J domain, this region, interacts, but the tail no longer binds. So this interaction of the C-terminal EVD tail of HSP70 only happens in class B and not in class A. So we have two things different between class A and class B. The first one is that class B have this GF inhibition that plays some regulatory role. And second, we have this new binding site between the C-terminal tail and the CTD1 only in class B and not in class A. But now the question is this interaction of any importance? I mean, it binds to the substrate binding domain, it's a disordered tail, maybe it's some mimicking of the substrate. So to see if this interaction is actually important for the function of DNA JB1, we tested the disaggregation activity of DNA JB1 together with HSP70 and HSP110 nucleotide exchange factor in disaggregating alpha-synuclein amyloid fibers. And we do that by monitoring the decrease in THT fluorescence. So THT is a dye that binds to the cross beta characteristic of the amyloids. And as the alpha-synuclein fibers are disaggregated, we can see the decrease in THT fluorescence. So this is with the wild type proteins. But when we repeat the experiment with HSP70, which is only lacking the 4 last amino acids, the EEVD tail, we see that the segregation activity is completely lost. And if we now repeat the binding and see the binding of DNA JB1 with this HSP70 lacking the C-terminal tail, we can now understand why there is no activity, because there is absolutely no binding between the two chaperones. And what was very surprising for us is that we didn't just lose the binding between this tail and the C-terminal domain 1, where the binding of the tail occurs. We also lost the binding of the J domain with this interface between the nuclear binding domain and the substrate binding domain, which is really far away from the tail. So now we were quite puzzled about why this disordered tail of the HSP70 is so important for the interaction. And to try to understand that, we decided to make a peptide which corresponds to the C-terminal tail of HSP70 and those EVD amino acids, and titrate this peptide into DNA JB1. So we did the titration, and you can see now that we add the peptide, we have binding, beautiful fast exchange binding. We can map all of those residues to the area in CTD1, mimicking completely the binding of the full-length HSP70. So we know that the CTD1, it's only the C-terminal tail of HSP70 that binds. But when we examined the spectrum closely, we saw that in addition to those beautiful fast exchanging peaks, we also have peaks in slow exchange. And all of those peaks in slow exchange are actually coming from the J domain. So when the C-terminal tail of HSP70 binds to CTD1, here, somehow the conformation of the J domain changes. And if we zoom in on this isoleucine 48, we can see what's going on. So the isoleucine 48 is very sensitive to the presence of this GF inhibition by helix 5. So if we record the spectrum of the construct that has the J domain GF and the J domain is inhibited, it will be here. If we record the spectrum of the free J domain without the GF inhibition, it will be here at about 13.5 ppm. In full-length DNA JB1, we can see that the J domain is completely inhibited. But when we added the peptide, we saw this new peak appearing, which correspond to the open conformation of the J domain. So when the C-terminal tail, the EVD tail, actually binds to CTD1, it somehow releases the GF inhibition of the J domain. But the binding occurs of the tail occurs here, and the GF inhibition is here. So we have two options. Either we have a really, really long allosteric network going across 100 or more amino acids from the CTD1 all the way to the J domain, releasing the inhibition. Or in fact, maybe the J domain itself, it's found much, much closer, somehow docked onto the almost docked onto the CTD1. And just to remind you again, we don't have a crystal structure of the full-length protein. This is just a model generated based on the structure of bacterial protein. So we wanted to test maybe the J domain in fact comes close in CTD1. And to do that, we used fermionic relaxation enhancement experiments. So we attach a spin label to the J domain. And we measured the intensity differences between the oxidized and the reduced state. And you can see here that the intensities of the J domain are of course reduced because we introduced the spin label there, but also we see quite a large reduction in CTD1. And if we measure the R2s and calculate the distances using the spin label, we can see that the residue 40, which we spin labeled, is found between 12 to 20 angstroms away from residues in CTD1. So in fact, the J domain is found really, really close to CTD1 in an interaction which we think is mediated by the GF region. And now I wanted to see, using the same PRE experiments, to see what happens when we add the C-terminal EVD tail. So when we added the peptide and repeated the PRE experiment, we saw that all those peaks that disappeared are back up again, meaning that the J domain no longer found close to CTD1. So when the C-terminal tail of HSP70 binds, it releases the GF inhibition, causing unfolding of helix 5 and freeing the J domain. So based on our NMR data, we now propose the following model for how class B J domain proteins interact with HSP70. So in the DNA JB1, we have this GF inhibition blocking the HSP70 binding site. And only when HSP70 binds, first for its C-terminal tail to CTD1, this causes steric interference that eventually causes the release of the J domain and unfolding of helix 5. And now this J domain is free to bind to HSP70. HSP70 will be activated. It will convert from ATP to ADP state and now bind substrates, which in our case are the amyloid fibers with high affinity and cause disaggregation. So this is for class B. For class A J domain proteins, we in fact see very, very different mechanism of binding. There is no second binding site in CTD1, and the J domain is constantly released, so it's always free to bind and activate HSP70. So in summary, we see that class A and B indeed interact with HSP70 from two very different mechanisms. But the question is this mechanism that we found that is present in DNA JB1 and this GF inhibition, is it really the reason that class B are the only ones that can function in protein disaggregation, and class A that lack this GF inhibition and the regulatory mechanism cannot? So in order to test that, we generated a mutant of DNA JB1, which doesn't have helix 5 and have a constantly released J domain. So we made the mutant by taking the residues of helix 5 of DNA JB1 and replacing that with residues from DNA J2 that doesn't have the GF inhibition. If we compare the NMR spectrum of the two, we see that they're very, very similar. The only differences are in the J domain and residues at this interface, because there's no longer docking of the J domain. And now, using this mutant, we can test if it's defective in protein disaggregation. So again, we're looking at the disaggregation of alpha-synuclein fibers with HSP70, nucleotide exchange factor HSP110. DNA J2 is completely inactive. DNA JB1 wild type is very nicely active. And when we test a DNA JB1 delta helix 5 that lacks the GF inhibition, we see that it's completely dead in the disaggregation. So in fact, this GF inhibition as regulatory mechanism is absolutely essential for protein disaggregation. And we can go further, we can make point mutations with various degrees of released J domain, and we can see as the percentage of the released J domain increases, the segregation ability of that DNA JB1 mutant decreases. So there is a new regulatory mechanism present only in class B J domain proteins, which is essential for protein disaggregation. In class A J domain proteins that lack this regulatory mechanism are in fact inactive in protein disaggregation. So if I have 2 minutes, I would like to briefly explain why is that. Because we found this mechanism and we understand that it exists and we understand that it's very important for protein disaggregation, but we were really puzzled by what is it that it's doing and why is it there. So I'll show you some of the more preliminary data. We teamed up with our collaborators, Gerd Buchau, that were working parallel to us and trying to understand the mechanism of protein disaggregation. And Anna Wintek, a postdoc in Bern's lab, actually now in the process of publishing her findings about how protein disaggregation works in terms of what is the function of each one of the chaperones. And what she found is that DNA JB1 chaperone will be the first one to be recruited to the fibers, and it will bind fibers with really high affinity. And then this DNA JB1 will recruit HSP70s to the fibers. Now, because DNA JB1 is a dimer, and we see that it has 4 binding sites for HSP70, it can recruit multiple HSP70s. Now, this presence of multiple HSP70s in vicinity to each other, this is from their work, is what will cause entropic pooling by HSP70s banging into each other and banging into the fiber walls, eventually breaking the fibers apart. And then we have the HSP 110 nucleotide exchange factor, which will release HSP 70 from the fibers. But Anna's work showed that actually, because HSP 110 is a really large 110 kilodalton protein, it cannot access well those clustered HSP 70s. So it only recycles the one that are non-clustered and non-efficient for disaggregation. So we have this beautiful mechanism of how the free proteins work in protein disaggregation. We wanted to see is to break it into steps and to see at which step our DNA JB1 delta helix 5 mutant that lacks the disaggregation will stop being functional. So we tested binding to the fibers. We see that all bind fibers the same. And then we tested recruitment to the fibers. So what we've done is we took fibers, pre-decorated already, bound already to the J domain proteins, and added HSP 70 and saw how much HSP 70 can precipitate together with the fibers. And we see that DNA JB1 recruits, wild type recruits a lot of HSP 70. DNA J2 does not do such a good job. So because class A, we think because class A only have the J domain binding site and lack the binding site in CTD1, they're not as efficient in recruiting, but the delta helix 5 looked almost as efficient as the wild type. So then the last thing that we tested was the clustering, this essential clustering of multiple HSP 70s in order to generate the entropic pooling forces. And this was an experiment done by Anne. She labeled half of the HSP 70s with acceptor, half of the HSP 70s with donor, and looked for fret. So when the two are near, will be high fret signal for the clustered, and non-clustered, there'll be low fret signal. So what you can see here, that only B1 wild type has high fret signal, A2 and delta helix 5 are not. So this, we think now that this GF inhibition and this regulatory mechanism actually needed to efficiently cluster HSP 70s on the fibers. And we think the reason for that is this architecture of DNA JB1 and the fact that HSP 70s first have to bind to CTD1 really, really close to the fiber wall in order to engage the fibers by being activated by binding to the J domain. So to summarize, we see that this GF inhibition is essential for protein disaggregation, and we think that it needed to ensure that HSP 70s are recruited to the proper position along the aggregate wall to result in efficient entropic pooling and disaggregation. We know that 2 binding sites per protomer are required for proper recruitment of HSP 70s to the amyloid fibers, but the presence of the 2 binding sites is not efficient for disaggregation. And in the grand scale of things, we see that 2 J-domain proteins that are structurally very, very similar actually interact with HSP 70 and activate HSP 70 for 2 different mechanisms. And we see that this presence of the GF inhibition that is only found in class B J-domain proteins is essential for the class B function, which is mainly protein disaggregation in the cell. And with that, I would end. I would like to thank 2 people from my lab, very talented postdocs from my lab, Ofer Faust and Meital Abraham, who carried all the experiments from our side. This was all done in collaboration from the group of Bernd Buchau, where Anne was the driving force. And I would also like to thank Neil London for his help with the Rosetta structure calculation. And thank you for listening.
Thank you, Professor, for an exciting talk and detailed mechanism of disaggregation. So the talk is now open for questions. Please raise hands if you have any questions. I don't see any hands. Yes, Ashutosh. Ashutosh, you can unmute yourself.
I have unmuted. So Reena, fantastic talk, Ashutosh here. So one question I have for alpha-synuclein disaggregation that you showed. So most of the probe that you showed is thioflavin T fluorescence, which actually probes the cross beta structure of the fiber. Now I'm curious to know if the HSP and DNJ releases this. So does it release a monomeric alpha-synuclein or oligomeric alpha-synuclein? What kind of alpha-synuclein is released? Because thioflavin T just gives you that it's not cross beta structure. So can you elaborate a little bit on that?
Yeah, absolutely. So you're absolutely correct. Thioflavin T is not a reporter of what what are the species that are released during disaggregation. Now, the fact that these proteins can disaggregate amyloid fibers, this is not our finding. This is Bernd Buchhaus' group showed it a couple of years ago. They see that there are monomer released for sure. There's a lot of monomers released, but they also see oligomers in all various sizes. So it's sort of a dynamic process where you have a release, both breaking of the fibers into smaller fragments and release from the ends.
So can I ask a follow-up question on the same topic? If the oligomers are released, does any of you or your collaborator did check that are these toxic? If they are toxic, then it's more dangerous than letting them be in the fibrillar state.
Absolutely. I mean, this is an ongoing question right now in the field. Are these chaperones disaggregation is actually good to the cell, or does it generate more seeds in order to promote disaggregation? The nice thing about it that we now know that only class B works. We know how to make it either work better or work less well. So once somebody figures out if we want chaperones to disaggregate or not, we can tell them how to either inhibit or activate the DNA JB1 disaggregation pathway.
Thank you very much.
Not clear. It's not clear yet.
Yeah, thank you very much.
So next question is by Grace. Grace, you can unmute yourself and ask.
Thank you. Nice talk. I was also wondering about the follow-up question of Ashutosh, I think the previous person. So I was wondering, what stage do you think these chaperones will come to the rescue of the substrate proteins? Is it known, or are you guys still solving it? That is my first question. And my second question is, if they are forming aggregates, they must be having like picomolar affinity, right? Generally, aggregates are prone to pull up every protein towards that. So is this affinity higher enough to rescue those substrates which are getting dragged into that aggregation pool?
Okay, so I'll start with the second question about the affinity, and then you'll have to remind me the first question. Sorry. So affinity-wise, it's amyloid fibers, so they don't co-aggregate with other proteins. The binding affinity of the J-domain proteins that recruit the 70s is a nanomolar affinity. And HSP70s are recruited there. They do their work. They bind with about 1 micromolar affinity, but their activity is enhanced by the fact that many HSP70s are recruited by DNA JB1 to the fiber. So there is an avidity effect and also the forces by entropic pulling to disaggregate.
Thank you. Yeah, my first question is like, at what stage do they go to the rescue?
Okay, so there are many different chaperones in the cell. They all, recent work, they all bind at different stages. So HSP70, which is the chaperone we mainly work with, it seems to be interacting with substrates across all stages, and which substrate will be recruited is dependent on which J-domain proteins it's working with. So it will work in binding to proteins already on the ribosome, or the moment they emerge from the ribosome to help them fold properly. It can bind misfolded proteins, preventing them from forming aggregates, so slowing down aggregation and refolding misfolded proteins. And together with DNA JB1, it can also work on fibers and disaggregate fibers and protein aggregates as well.
Thank you.
So I think one last question from Samrat. Samrat, you can unmute yourself.
Hello, ma'am. Nice talk. My question that Ashutosh sir already asked, but I would like to ask that again, whether when these protein aggregates are formed, is there any trigger mechanism which triggers this domain for going to the rescue of the protein as well? Are these protein any sequence specific, as protein will be aggregating in multiple, means there will be dimerization, oligomerization. So how this domain exactly know where to bind and where to cleave? And after cleavage, is there any, means after cleavage, do you get active form of protein or not?
Okay, so it's not a cleavage because the protein is, I mean, alpha-synuclein monomers are really small.
Okay.
The aggregation core of alpha-synuclein is known, not our work. It's been known for many people's work. Where the chaperones bind on the fibers, this is something that Bernd Buchau's work showed, and it's going to be published soon. So we know where on the fibers the chaperones bind. Once they bind, they break the amyloids, and they break them into smaller oligomers, smaller fragments, or to monomers. Once the monomers are functional. And it's the same from protein disaggregation. So the same machinery can disaggregate amorphous aggregates, and there we can test activity of enzymes like luciferase or many other model substrates, and the proteins are completely active.
Okay. Thank you, ma'am.
Okay, so if there are no further questions, let us thank Professor Rosenzweig for a beautiful talk, and today's speaker, the other speaker, Dr. Sheetal Jain. Over to you, Professor Suryaprakash.
Okay, Keith, can you please display next week's poster?
Yes. So next week we have two more exciting talks coming up from Professor Francesca Marassi and Professor Pradeep Bhattacharya. So stay tuned in for 29th September.
Okay. So thank you, Keith, for conducting this, sharing this session. And once again, I would like to take the opportunity to thank the speakers, both Reena and Sheetal, for their exciting talks. We'll meet again next week at the same time. So thank you. We'll call it. Thank you.
Thank you.